This video is part 2 of non-Mendelian inheritance and will cover epigenetic inheritance. Recall that epigenetic inheritance breaks Rule 2 of Mendelian inheritance from the previous video in that genes are not passed along unaltered, at least in terms of alterations affecting their expression. Epigenetic inheritance refers to a pattern in which a gene's expression is modified. This is not a modification of the actual DNA sequence and also not a permanent change. Two common mechanisms are DNA methylation and chromatin remodeling. These changes often occur in the formation of sperm or eggs, or in some cases in early embryonic development. One main reason for these epigenetic effects is to regulate gene dose. That is to make sure each organism has the correct number of active genes. We see this played out on a chromosome scale with the X chromosome. So let's examine that as an example. X chromosome inactivation, or X inactivation for short, is the process by which one of the X chromosomes is inactivated and compressed into a tightly coiled heterochromatin ball called a bar body. The choice of which X is inactivated is random. X inactivation makes sure that there is only one active X chromosome in each cell. So this only happens in females, because males only have one X chromosome to begin with. Additionally, this pattern, which is to say which X chromosome is inactivated, is maintained in all subsequent cell divisions. So before we look at an example of inheritance patterns, let's step back and ask why we need dosage compensation in the first place. Our X chromosomes contain many genes that encode for proteins that we need for basic organism function. Since the basic metabolic needs of males and females is largely the same, there's no reason that females would need double the copies of X chromosome genes compared to males. Thus, X inactivation regulates the gene dosage in females so that although they technically have two copies of each X chromosome gene, only one is actually active in any particular cell. Now that we understand the why a little better, let's look at an inheritance example. Calico cats are an example of a phenotype that is due to X inactivation. The gene for orange or black fur is located on the X chromosome. Male cats can be either orange or black depending on which allele they inherit. However, female cats can be orange, black, or calico. The calico phenotype is caused by being a heterozygote for the gene that controls this coat color. Different cells will have different X chromosomes inactivated, so some cells will express the allele for the orange coat, and others will express the allele for black coat. This results in the patchy fur color you see in calico cats and explains why calico cats are always female. There is another form of epigenetic inheritance called genomic imprinting. This inheritance pattern is typically due to methylation, which stops the expression of a particular gene in either the maternal or paternal copy of the gene. This results in the offspring expressing either the maternal or paternal allele, but not both. As discussed previously, methylation affects gene expression when it binds to regions that affect the binding of transcription factors. So let's quickly look at an example. Here we have two syndromes that are both due to small deletions in chromosome 15. Whether you show Prader-Willi syndrome or Angelman syndrome depends on which parent you inherit the deletion from. This diagram shows this pattern. Each of these syndromes is due to the lack of expression of specific genes in this region of chromosome. These genes typically methylated in one of the parents, so when the region with those genes is missing in one parent, the copy from the other parent is not expressed to compensate. So when the father passes along the deletion in chromosome 15, genes like SNRNP and NDN are not expressed since the maternal copy is silenced. This results in Prader-Willi syndrome. In contrast, when the mother passes along the deletion in chromosome 15, the gene UBE3A is not expressed because this gene is silenced in the paternal copy of chromosome 15 due to methylation. This results in Angelman syndrome.